The spark of life electricity in our cells. Our bodies are electric. We don't mean this figuratively, we mean this literally. Every single one of our cells possesses an electrical charge, a tiny voltage difference across its membrane. This is not some strange quirk of biology. It's fundamental to life itself. This electrical potential, called the resting membrane potential, is the foundation for nerve impulses, muscle contractions, and even the beating of our hearts. Without it, we wouldn't be able to think, feel, or move. Imagine the vast network of electrical wiring in your home. It allows you to power lights, appliances, and electronics. Similarly, the intricate network of cells in our bodies relies on electrical signals for communication and coordination. These signals, known as nerve impulses or action potentials, are sudden, rapid changes in the electrical potential across a cell's membrane. They are the language of our nervous system, transmitting information from our senses to our brains and back again. But how does this cellular electricity work? What generates these electrical potentials and allows them to travel throughout our bodies? The answer lies in the remarkable properties of cell membranes and the tiny charged particles called ions that constantly move across them. Join us as we delve into the fascinating world of cellular electricity, exploring the concepts of resting membrane potential and action potentials. We'll uncover how these fundamental processes underpin the very essence of life, allowing our bodies to function with such remarkable precision and complexity. Every cell in our body acts like a tiny battery, maintaining a difference in electrical charge across its membrane. This difference, known as the resting membrane potential, is typically around -70 millivolts. The negative sign indicates that the inside of the cell is negatively charged relative to the outside. Think of it like a car battery. A car battery has a positive and negative terminal, creating a potential difference that can power the car. Similarly, a cell has a separation of charge across its membrane, creating a potential difference that is essential for its function. But how does this separation of charge arise? The answer lies in the uneven distribution of ions, or charged particles, inside and outside the cell. The cell membrane, which acts as a barrier, is selectively permeable, meaning it allows some ions to pass through more easily than others. Specifically, the cell membrane is more permeable to potassium ions than to sodium ions. Inside the cell, there is a high concentration of potassium ions, while outside the cell there is a high concentration of sodium ions. This difference in concentration creates a concentration gradient, a force that drives the movement of ions from an area of high concentration to an area of low concentration. The cell membrane is not just a passive barrier, it is studded with specialized proteins called ion channels that act as gatekeepers, regulating the flow of ions into and out of the cell. These channels are selectively permeable, meaning they allow only specific types of ions to pass through. Think of these ion channels as doorways in a wall. Just like doorways regulate the flow of people in and out of a room, ion channels regulate the flow of ions in and out of the cell. Some ion channels are always open, allowing a constant trickle of ions to pass through, while others are gated, meaning they open and close in response to specific signals. Among the most important ion channels are potassium channels, which allow potassium ions to flow out of the cell, and sodium channels, which allow sodium ions to flow into the cell. The movement of these ions is driven by both the concentration gradient and the electrical potential difference across the membrane. Remember that the inside of the cell is negatively charged relative to the outside. This negative charge attracts positively charged ions like potassium and sodium. However, the concentration gradient for potassium is outward, meaning there is a greater tendency for potassium ions to move out of the cell than into it. The opposite is true for sodium ions. The concentration gradient for sodium is inward. The movement of ions through ion channels would eventually equalize the concentration of ions inside and outside the cell, eliminating the resting membrane potential. However, the cell has a remarkable mechanism for maintaining this potential, the sodium-potassium pump. This molecular pump embedded in the cell membrane constantly works to pump sodium ions out of the cell and potassium ions into the cell. It does so against their concentration gradients, meaning it requires energy to operate. This energy is supplied by ATP, the cell's primary energy currency. The sodium-potassium pump is essential for maintaining the resting membrane potential because it counteracts the leakage of ions through ion channels. For every 3 sodium ions pumped out of the cell, 2 potassium ions are pumped in. This unequal exchange of ions helps to maintain the negative charge inside the cell. The sodium-potassium pump is a prime example of how cells expend energy to maintain order and stability. Without this constant pumping action, our cells would be unable to maintain the electrical potential difference that is essential for their function.